Yeah. Yeah. Good. How are you? Yeah, probably five. Yeah. Yeah. Webinar. No, I don't.
Oh, I guess. I have been. Did you come up for no, but you get to see that. Okay, I've been over there just days. And so, you know, it's pretty awesome. Yeah, a lot of spaces over there. Uh, I think it was David and I were going there for lunch or something like that. I had no opinions for like 8 years. Yeah. But I know, I think one of the people are divided. So, yeah. Yeah, so nice opportunity. So got it, yeah. So cool. So yeah. Oh, yeah, we need to wait. Okay, so, oh, I guess I should use the mic for Zoom people. Did you say that? Okay, Zoom, I hope that you can hear me too. Welcome everybody. Oh, great. We got awesome. So welcome everyone to Rachael Aber's PhD defense talk. I'm Ben Diel. I'm Rachael's advisor in the Integrative Biology department. And I'm joined by Katie McLaughlin, who is Rachael's advisor in the statistics department. So the run of show is going to be that we're going to have a 45-minute talk from Rachael. We're going to have about 10 minutes for questions from the audience. And then we'll go to the private portion of the defense where the committee meets with Rachael for further discussion. We anticipate that will take about 2 hours. And so the whole thing will wrap up 11:30 to noonish. So I'm going to take a few minutes now, or probably just a couple minutes, and introduce Rachael, and then I'll pass the mic to Katie, who will say a few more words. So Rachael got her undergrad degree in biology from the University of Portland. And while she was there, she did research in acoustic ecology in marine systems. And she won several awards while she was an undergrad, including for outstanding research. She joined our lab in the fall of 2018 with continuing interests in ecological dynamics and data analysis. And she brought those interests to data sets that we were looking at and that she helped collect in epidemiology and public health. While here, she has won several more awards, including an ACM SIG-HPC Computational and Data Science Fellowship, ARCS Fellowship, and a Provost's Distinguished Fellowship. And today she is defending a dual PhD in integrative biology and statistics. And if you're wondering if that is more work than a single PhD, I would say from the outside that the answer is definitely yes. So it's a really big achievement. So next month, she's starting a job as a research scientist at Exponent, which is one of the leading consulting firms in the US. She got that job because when she presented some of her work at a conference a few months ago, they recruited her. And she's going to be presenting some of that work today on a new method that she developed for analyzing count data. So when I think about Rachael's qualities, the words that come to mind are kindness, integrity, precision of thought, and relentlessness in her pursuit of good science. And these are, of course, exactly the traits that a lot of us in the business aspire to. And so I'm not surprised at all, Rachael, that you were offered a job before you had defended. And you have led by example in the lab and just by sheer technical ability as well. And we're going to miss you a lot. And we can't wait to see what you get up to next. I'll just pass it to Katie.
Yeah,
Do you want to do the mic? Zoom?
All right. Hi, everyone. I don't want to take up too much more time because I know you're all here to hear Rachael, right? And not hear me and Ben. But I just want to echo what Ben said and say that it's been such a joy teaching and advising Rachael. And so we were so happy when she decided that she also wanted to pursue statistics coursework and a dual degree on top of her already ambitious IB research goals. So over the years that I've worked with Rachael, I've come to learn that she has a great intuition for statistical concepts and theories, and the unique perspective that she brings from her multidisciplinary background really allows her to push the boundaries of interesting research questions and new approaches. And so in addition to all of those great qualities that Ben listed, The two things that have really stood out to me are Rachel's independence and her curiosity as a researcher. And that combination has made my job as an advisor very easy because she always has great ideas about how to tackle new problems, and that will serve her very well in her future work. So without taking up much more time, here's Rachael, and I cannot wait for her to share her research with you.
All right. Thanks, Ben and Katie, for those kind words. I will miss you both too. All right, so I'm excited to talk to you today about my dissertation on quantifying and predicting heterogeneity in infectious disease spread. So a quick roadmap of where we're going today. First, I'll talk about why I study heterogeneity in infectious disease spread in the first place. I'll then spend the bulk of the talk talking about my first chapter, which is time series modeling of epidemics in complex populations. And then I will spend the remainder of the time talking about my other two chapters, the impact of commuter flow on SARS-CoV-2 dispersal in Oregon. So that's the virus that causes COVID-19. And the effects of population structure and healthcare heterogeneity on novel pathogen detection. So why study heterogeneity in infectious disease spread? One reason is that metrics that capture heterogeneity can contribute to pandemic preparedness. So as we're all aware, infectious diseases are a persistent global health burden, in part via novel pathogen emergence. It was found that about 335 novel pathogens emerged between 1940 and 2004. And the rate has only been increasing with the interplay between land use change and climate change causing clusters of spillovers. So, and spillover is essentially just when a virus or another pathogen moves from an animal host into humans. So it's necessary to quantify the properties of the resulting epidemics. For example, you may have heard of R0. That's the basic reproduction number. So that's measuring in a completely susceptible population how many secondary cases on average are caused by an infected person. It's also necessary to quantify the human systems that propagate and monitor these pathogens in order to inform public health policy. So one important aspect of any epidemic is transmission heterogeneity and the resulting heterogeneities in spread patterns and discrepancies in how a surveillance system might be able to detect a pathogen or the pathogen in question. And so the type of heterogeneity that I'm interested in is depicted on the right here. So it's what's called structured heterogeneity, whereas the middle can be considered more noise. So the quantification of these aspects of an epidemic ensures efficient allocation of resources. For example, quantifying transmission heterogeneity can infer or can inform where and when surge capacity is needed. So just briefly, my PhD chapters focus on disease spread heterogeneity in the following ways. So my first chapter uses widely available case count data to gain a predictive understanding of transmission heterogeneity. So that's essentially variability in the number of people that each infected individual goes on to infect. So yeah, you might have heard of super spreading, which is just the refers to elevated transmission heterogeneity and has been more used in popular media. So my second chapter then models population level spatial heterogeneity in SARS-CoV-2 spread using genomic data. And my last chapter incorporates population structure via factors like incidence heterogeneity to model the probability that a novel pathogen is detected. So jumping into my first chapter, time series modeling of epidemics in complex populations. So using time series to understand epidemic trajectories is nothing new. However, metrics of variability are somewhat overlooked. And one can ask questions like Graham et al. Asked, which is how is variability related to different phases of an epidemic? So they were interested in measles elimination and found that as a location, country, or a region approaches elimination of the pathogen, their time series follows this arc where the mean drops off and the interannual coefficient of variation increases. So then what they were able to do is take countries or locations and plot them along a path and kind of figure out where on the path to elimination they were. Inference methods based specifically on the dispersion of epidemic time series are emerging. For example, an index of effective aggregate dispersion was recently proposed to measure or elucidate clusters of infection directly from incidence data. And heuristically, you can think about dispersion as when many of the disease cases occur in narrow windows of a time series. And this can suggest important underlying biological processes like transmission heterogeneity or changes in the reproduction number of a pathogen R. So I mentioned R naught earlier. That's just the R at the beginning of an epidemic. So it was found that COVID-19 transmission heterogeneity increased over time in Hong Kong, and also as new variants emerged. And so what we wanted to do was use incidence data to see if we could detect signatures of elevated transmission heterogeneity in US counties. And so a little bit more on dispersion and its relationship to case clustering or crowding. So dispersion and the dispersion parameter are part of a negative binomial framework in this model that we created that models variance in incidence at a time point more flexibly than if you used a Poisson model. And so one way to think about the dispersion parameter in the context of a time series model of counts is that events are 1 + 1 over the dispersion parameter more crowded in time relative to a Poisson model with the same mean. And so a mean crowding parameter was initially proposed in an ecology context, which was it's the mean number per individual of other individuals in a quadrat. So you can think of it as the number experienced by an individual. And so the degree of dispersion is also the degree of crowding. Here's the relationship between the two, or they're related in this way. So mu star is the mean crowding, and theta going forward is the dispersion parameter. So just a quick note that high dispersion corresponds to low values of the dispersion parameter. I didn't name it, but that is just something to try and keep in mind as we go forward. And so this dispersion or mean crowding framework is a useful way to think about clustering in case count time series. So instead of thinking about an animal in a quadrat experiencing other animals, you can think about an infectious individual experiencing other infectious individuals in their local spatiotemporal neighborhood. So on the left, you can see a super spreading pathogen. And so in the center, that infected individual is experiencing many other infected individuals in their local neighborhood. And so that's what I mean by high dispersion or high crowding, and clustering as well. So you can think of those as clustered. So standard theory proposed that incidence at a time point can be modeled by a negative binomial random variable with the mean equal to the epidemic intensity. So this is just a special kind of moving average. It's the expected number of cases at a time point, and dispersion equal to the previous incidence. And I'm not going to go through the full model derivation, but the derivation comes from the fact that disease spread forms kind of a tree, like a phylogeny, where all infections are come from infections in the previous generation. However, this model assumes that the transmission rates across those lineages are independent and identically distributed. And there's reason to think that this assumption is violated, and other assumptions of the model that I want to talk about are violated. Partly because of what I discussed earlier, that some individuals may transmit the disease to more than others. And so to investigate where and when this assumption or set of assumptions is violated, we quantified how the dispersion parameter changes over time and to what degree it's actually governed by incidence. To model the mean, mu, we used a linear predictor that includes a natural spline in time to account for autocorrelation between counts. And we adjusted for population size using an offset in the model. So for those who aren't familiar with that term, I just added a few quick lines of algebra. So instead of modeling the expectation of counts at time I, it's modeling the expectation of counts per individual. And then through some algebra on lines 4 and 5, that log of n sub I pops over to the right-hand side. And so at a time point, incidence follows this distribution where mu is modeled using that linear predictor from the previous slide. And then I've also just added the expectation and variance here to highlight one more time that high variance and high dispersion are actually low values of theta, just before we get into the results. And so what we did was fit this model on a rolling basis to each time series in the data set. And so what that means is we fit the model to a window of data and then got one estimate of the dispersion parameter and then started sliding that window down the time series so that we ended up with a series of dispersion parameter estimates for each time series. And then instead of just fitting the model at each time step, we developed a fairly straightforward likelihood ratio test. And so what that involves is that at each time step, a null model or restricted model, you might be familiar with that term, is that does not allow theta to change. And then a flexible model that allows theta to change at a midpoint is also fit. And then a likelihood ratio test is conducted. You get a p-value from that. And then as you slide the likelihood ratio test procedure down, you could get a p-value for each time point. And that indicates, so a small p-value in that sense would be evidence in favor of a change in the dispersion parameter. And so to test the validity and power of this approach, I created a simulated time series data set where there were both Gaussian and uniform epidemic curves with an attack rate of 0.1. So what that means is just that the proportion of people who were infected by the end was 0.1. I varied the magnitude of the theta change, the location of the change within the epidemic curve, the underlying population size, and the curve shape, as I mentioned above, and applied the likelihood ratio test to each epidemic curve at its known change point in theta. And so I just added a visual of, oh, and I should say too, that this is just an example of one of the Gaussian epidemic curves with a change point in theta, an increase in theta, so decrease in dispersion at the midpoint. And it's a little bit more clear here. So in C, that's the constant dispersion parameter used in generation of A. And you can see that the cases in A are consistently volatile. And then in D, there's a changing dispersion parameter. It increases, and then you see the volatility drop off in the second half. So keep this O and X in mind because they are here, where E is the all time series with an underlying population size of 50,000. This was the smallest population size that we wanted to examine in the empirical data. And then F is all time series with an underlying population size of 10 million. And so we were looking for approximately those characteristic power curves where when there's no effect, the average p-value is around 0.5, and then it drops off as the effect size increases. And so then we went on to apply this method to weekly case counts from US counties between the beginning of 2020 and early 2023. And we estimated theta t for each of 154 time steps for 144 US counties using an iteratively reweighted least squares approach. And the 144 counties, those are just the largest 3 counties in each state. And so here's an example from the empirical data where you can see that a change in volatility of counts, especially if you draw your attention to the start, near the start of 2022, that corresponds to this kind of trough in the dispersion parameter. And then I've also colored the dispersion parameter estimates by the corresponding likelihood ratio test statistic value. So you can see that red kind of denotes a change in the regime of the dispersion parameter. That's high values of the test statistic. And so now here are the results for all of the counties examined. On the y-axis, that's the 144 counties, and then time is on the x-axis. So each time series is one of the horizontal lines. And so you can see that high values of cases, which is darker, deeper blues, so higher incidence, corresponds or is kind of flanked by high dispersion, which is low values in yellow of the dispersion parameter, or sorry, high dispersion, yeah, is low values of the dispersion parameter in lighter yellows. And so here in A, that's just another way of visualizing this panel marked B. So these are binned estimates of the dispersion parameter with the yellow bin denoting the most dispersed category. So you can see that highly over dispersed patterns are observed near where peak incidence occurs, and also near the end of the time series. You see that little yellow increase, and that is consistent with more heterogeneity in reporting, susceptibility, and transmission. And then unsurprisingly for the counties, there's also consistently low p-values denoting a change in theta, or a putative change in theta, right around where the dispersion parameter changes. And so in conclusion, the likelihood ratio test procedure developed performed well for the relevant range of population sizes, 50,000 to 10 million. And we found that dispersion is high at unexpected times. So instead of being high at peak incidence as predicted by the model that I showed you earlier, it's high near peak incidence, so kind of on the way up to and down from the peak magnitude of an epidemic. And one reason this could be is that as we approach the kind of peak magnitude of an epidemic, some people might be really prone to adhere to social distancing measures. They might be really concerned, and then others may not. So there might be a lot of heterogeneity in adherence. And also found that, unsurprisingly, the concentration of changes in the dispersion parameter indicated by the test occurs right around that time. And so these regimes of the COVID-19 pandemic that were subject to increased dispersion, these could be candidates for increased surge capacity once potentially reporting error is accounted for as well. Okay, so now I'm going to shift gears, and instead of talking about transmission heterogeneity. I'm going to talk about spatial spread patterns. So modeling the spatial spread of infectious pathogens is important due to spatial differences in susceptibility to different variants, and because there may be impacts of local control and mitigation on overall epidemic trajectories if connectivity between regions is strong. So one way to model spatial spread is to use spatially indexed viral genomes in phylogeographic methods. So these methods aim to model the processes that determine the spatial distribution of lineages. So here is a reconstructed phylogeny of the SARS-CoV-2 spike protein from Oregon data between 2020 or beginning of 2020 to early 2023. And so you can see that out of these models, these phylogeographic models, you get a reconstructed phylogeny, but you also get what's called transition rates. So here, the counties, the legend is not showing up that well, but these are county names in Oregon. And so you can see the transitioning between counties is also a product of these models. So you get out an estimated history of where the pathogen's been. So I wanted to investigate the predictors of these transition rates or model these transition rates themselves. And the reason I wanted to do this is because predictions of influenza spread were found to be most accurate when human mobility data was used alongside the spatial information that's already inherent in spatially indexed genomes. So because the spatial heterogeneity of an epidemic is tied to behaviors that create patterns of exposure like air travel or contact at restaurants, I hypothesized that commuter flow, so workplace to home flows in Oregon, were associated with SARS-CoV-2 spread during a portion of the pandemic. And so I tested this hypothesis at two spatial scales. One was at the county level, and then I also grouped counties together using a community detection algorithm, the walk trap algorithm. And so it produced modules that are densely connected in terms of their commuting patterns, and therefore likely in terms of their contact patterns. And so the structure of the communities reflects a high degree of commuting flow to and from Portland. So you can see that many counties around Portland were grouped together based on their commuting flow. And then also a couple in northeast Oregon. And then the black lines denote, the thickness of the black lines denotes the magnitude of flow between regions. And so how does one fit a model that both infers a phylogeny and transition rates and also models those transition rates? One way is by using, or the only way that I know of is using Bayesian inference. So BEAST specifically produces a posterior distribution on both the quantities that dictate the reconstruction of the tree and on the GLM coefficient that indicates how commuter flow is related to viral transitioning. So don't worry too much about the specific terms, but I'll just go through a few of them. The horizontal bars denote the data, which is a multiple sequence alignment. So it's a posterior of the evolutionary parameters and the GLM parameters given a sequence alignment of spike proteins in our case. And then the prior distributions that come after the likelihood, those are mostly chosen based on knowledge of the pathogen and other things. So if you, for sake of time, I'll keep going, but if you have more questions about this at the end, I'm happy to talk about it more. So we found that commuter flow is an important predictor of SARS-CoV-2 spread. So the posterior inclusion probabilities of this predictor were 1 in all sequence down samples in the county level case, and then over 0.99 at the community scale. And the actual coefficient estimates indicate that an increase of 1 unit in standardized commuter flow corresponds to about a multiplicative change of about 2.25 in viral spread in the community case and 2.24 in the county case. And so this implies that commuter flow is predictive in a way of SARS-CoV-2 spread, and maybe intervening at key points commuter wise might help slow spread of future pathogens or future pandemics. All right. So now I will talk about my last chapter, the effects of population structure and healthcare heterogeneity on novel pathogen detection. So the probability that a novel influenza virus is detected can be most simply modeled using a framework that just considers the probability of detection of a given case and the total population size n. And so in this chapter, I'm interested in surveillance system failure, which is essentially when the number of infected cases in a population surpasses some threshold or a given threshold without any of those cases being detected. And so that surveillance system failure is just essentially, if you're using the model above, it is just that same equation 9 without the 1 minus in the front. So that's the failure probability. Just pause for a second so folks can see that. Okay, so in this model, I was interested in relaxing the assumption that each person has an equal probability, or each given case has an equal probability of being detected. And the reason for that is that this p-detect quantity depends on the case seeking care and also the case being tested. And we know that these quantities vary systematically. So, for example, there's documented medical mistrust among Native American populations. And there's also a link between mistrust and hesitating to seek care. So that's one reason why p-detect might vary systematically. And then also there were found to be rural-urban disparities in testing, which again also affects the detection probability. And then I was also interested in relaxing the equal incidence assumption. So that's from this, that I is the probability of infection for each person. And that might not be equal for everyone because it was found, for example, that there's a higher risk of COVID-19 infection associated with residing in a neighborhood with financial insecurity. So again, this might vary systematically over Oregon and other places. And so I used the total population size from Oregon in the previous project and set the number of cases of the novel pathogen to be 500 because outbreaks larger than that are unlikely to be contained. And I took into account the connectivity between regions in Oregon to decompose the overall non-detection probability. And so in these following model extensions, I incorporate subpopulation structure via incidence heterogeneity and detection probability heterogeneity, and then also use actual Oregon subpopulation sizes to try and reduce the bias in estimating the overall surveillance system function. And so I created one model extension, which assumes that there's emergence of a variant of an already circulating pathogen. So the probability of system failure or non-detection is just the product over the K regions of the failure to detect the pathogen, where the local incidence estimate is, it relies on you having like a stationary distribution for a similar pathogen. So X sub I is the probability that the pathogen will be in that region I, and C is the total number of cases, and then N sub I is the population size of the ith region. And then I also created an outbreak model, which assumes that instead the outbreak is from a spillover and therefore occurs in one and only one of the regions. So the probability of failure is just decomposed according to the law of total probability, where the estimate of the outbreak occurring in region 1 is N1, which is the population size there, over N, the total population size of Oregon. And then the second term that it's multiplied by is the conditional probability that the system fails given that the outbreak occurs in region 1 and only region 1. And then I1 is a local incidence estimate in region 1. And so through simulation of these three models, the first row is using equal population sizes, and then second row is using actual Oregon subpopulation sizes of the regions. Model A, or sorry, the red denotes the original model that we saw on the first slide of this. B denotes the, blue denotes the endemic model, and green is the outbreak model, where again, the outbreak is just occurring in one of the regions. And so we found that when we assume, or when we use the original model and the situation is actually the outbreak model, we might be producing anti-conservative estimates of non-detection probability. So we may be overestimating how well the detection system is performing. And then within the green model, the outbreak model, I investigated the probability that, or I investigated the non-detection probabilities if the probability that the case is detected, a given case is detected, is correlated or negatively correlated with the probability that the outbreak occurs in that region. So for example, if the probability of outbreak in region 1 is large and the probability of detection for the cases in region 1 is small. So I found that when these quantities are negatively correlated, the detection system performs the worst. So these colors on this plot are the pink is the mean of the detection probabilities for that simulated correlation, and then black is 75th percentile, and the 95th percentile there is in green. And so unfortunately, this is a somewhat realistic assumption because, and the reason that I looked into this is because rural areas where there's the highest degree of agricultural operations are at most risk of spillover events, and they are also the most under-resourced when it comes to testing. So in actuality, we might not be at that correlation 0 place. We might be more towards the negative correlation part of the plot. And so in conclusion of this chapter, infectious disease surveillance systems perform better at monitoring pathogens that are already widespread and just a variant emerges. And they're most at risk of failing to detect a completely novel virus that results from a spillover, even when the number of cases in both scenarios is identical. Okay, so in conclusion of this dissertation talk, some high-level points just to take away. I found that high case count dispersion doesn't occur at peak incidence as predicted by standard theory, but instead it occurs near peak incidence. I found that spatial heterogeneity and spread is somewhat predictable using commuting data, and also that failing to account for heterogeneity and correlation in detection systems may lead to us overestimating how well they're doing, and therefore leading to maybe a false sense of security in terms of resource allocation. And so I just wanted to take the last few minutes to say a lot of thank yous because I could not have done this without all of you. Thank you to my two very brilliant co-advisors. It's been such an honor working with you both. Thank you so much to the statistics community. I could say all of your names and have something great to say about all of you, but specifically, thank you to Gowrie and Ruby for the long walks and supporting me. And thank you to Sarah Emerson and Duo Jiang for helping me realize that advanced degrees in statistics were possible for me. And thank you to my IB community, specifically my cohort. I was so lucky to go through the pandemic with your support and living with some of you. Thank you to members of my lab, Isak, Liz, and Noah. I am really grateful, Isak, for your friendship. And Liz and Noah, I miss you and thank you for helping me find my feet at OSU. Thank you to my friends outside of OSU for still being my friend, even though I invested so much time in this. Thank you to my wonderful family, mom, dad, Mike, and Annika. Thanks for always cheering me on. And last but not least, thank you to my wonderful partner, Zach, for believing in me always, even when I didn't believe in myself. And here are my references. If anyone is interested in that. Thanks.
Have lots of time for questions.
All right, what parts of the following statement are false? I think this is what I'm taking from your whole thesis, and so I want to know what I misunderstood. So pandemics or diseases are most likely to be missed and therefore most likely to occur in rural areas, but they're most likely to spread or become pandemics in cities because it's the most likely to get to the cities because that's where the most commuter flow is happening. And that's where the population sizes are largest, and therefore we're more likely to get super spreading or sorry, high heterogeneity. Yeah, in the right before maximum size.
Yeah, so I think that's right in part. The only way I'd amend that is spillover is most likely in rural areas, so you're right about that. And then if a rural area is very connected to an urban area via commuter flow or another means, that could be bad news in terms of if it was right before like a time when super spreading was occurring a lot. But keep in mind, we didn't look at the dynamics of smaller counties in that first chapter. So that's a bit of an unknown too.
So
Yeah, when it comes to scale, so you talk about having like local, regional, and like country scale. If I were a pathogen, like what would be my preferred scale to go to? Like having that structure in the community. So like what is from a different perspective, not just like if I were a pathogen to survive in a community. What would be like the main key resource?
Oh, probably susceptible people, right? So I would say any kind of area where there were enough people that hadn't been infected before. Yeah.
In your third chapter, I think one of the models, the second model, can you talk a little more about what the incidence estimator is?
Yeah, so like I1. So that essentially because we're assuming that the outbreak occurs in one region and only one region, that's just simply the number of cases C over the population size in that one region.
Noah says, great job, Rachael, for wonderful science.
Thanks, Noah.
other questions from the audience?
Yeah, I have one more. I'm still trying to understand why crowding is. Why heterogeneity is maximized right before the peak and right after. You addressed it, but it's not sunk in for me yet. Tell me your mechanism for that again.
But like why it's,
Over time, so you know, the heterogeneity was maximized. Not at the inflection point, but you said from prior to the peak, like an
Mhm.
early warning almost. Then right after the peak, so
Mhm.
blanking it.
yeah.
I assume there are different mechanisms. I think you said there were different mechanisms that you hypothesized, but could you explain those again?
Yeah, so one, I guess it's still kind of an unknown, but one hypothesis is that around peak incidence, there's more variability in how people adhere to social distancing guidelines. So around that time might be when some people really buckle down and stay inside. And then that also might be the time where people who are not going to adhere are going out and creating like bigger clusters of infection.
The social behaviors. Sure. Thank you.
No problem.
Hi, Rachel. Outstanding, congratulations. I'm an atmospheric scientist, so I'm completely ignorant of much of this. But how, are you able to, incorporate mitigation measures? So let's say you're advising community or government, and they would want to know about steps that they can take to mitigate. Are you able to maneuver the model in ways that you can have a predictive capability? You know, so they may incorporate some change in public policy, X number of riders on buses or something. How close are you able to take what you have in this more of a monitoring surveillance type scenario and incorporate those changes and have it predictive?
In the third, I'm assuming you mean in the third, the models from the third chapter. Okay, so it would be these. I suppose it's a little difficult to use them in a predictive sense. But in terms of it, because it's more about failure to detect and probability. So maybe it's more useful in creating hypotheticals. So say you have some kind of conjecture about, for example, the stationary distribution of a pathogen over space. So say you assume that it's most likely to be in X area, then what would be our detection probability if like the P detect varied in these ways or that way. So yeah, I'd say it could be used in prediction, but more of a hacky way of just simulating different outcomes.
Okay, well,
Okay.
if there's no more questions, thank you all for being here, and let's thank Rachael again for. So,